intratumoral immunotherapy is to basically use immunostimulatory products and deliver them directly into the tumor in order to use the tumor as its own vaccine. That's the whole paradigm, and the term of intratumoral immunotherapy is also used in comparison to in situ immunization, because the aim is really around the immunization against the tumor. It's not a very novel concept per se, because it has been tested in the past. The interest was mostly in the academic sites and academic investigators, but the novelty is that with the recent revolution of immunotherapy, we have now plenty of biotechs and big pharma that are now interested by developing immunostimulatory products to be injected directly locally into the tumor, with two aims: have more potent immunostimulatory drugs and less toxicity for patients. We have preliminary results about activity of intratumoral immunotherapy. We have data with oncolytic viruses, where we know that you can generate abscopal responses upon local injections, which means that you can inject a couple of tumor lesions and still generate an immune response against distant non-injected lesions and have a systemic effect of your local therapy. We also have data with synthetic molecules, not virus, such as toll-like receptor agonists, for instance, that have been tested in lymphoma, B-cell lymphoma and T-cell lymphoma, where again we have been able to see abscopal responses, or responses in non-injected lesions. So these are preliminary data in humans, and we have now coming data of combination of local immunotherapy together with systemic immunotherapy, with interesting early data showing that you can overcome the resistance to an anti-PD-1 failure, for instance, by providing a local immunostimulation together with a systemic additional immune checkpoint antibodies. This therapeutic strategy raises many practical questions in terms of clinical development of these compounds. We are completely in a different mindset than systemic delivery of dose, where when you do a phase 1 trial, you're looking for the maximum tolerated dose. When you do local treatment, the aim is not to have a maximum tolerated dose, it's to have a local bioactive concentration of your product. And so then comes the questions of what is the dose intensity that you need to generate to get this immune activation locally. So many questions are coming in terms of what dose should you adapt to the volume of the lesion, should you adapt to the number of lesions that are injected. All these questions are still pending, and we don't have practical answers to that. We will have an ESMO sponsored consensus meeting tomorrow with people from the academic side, people from the industry side, to brainstorm around these questions and have clear guidelines on how we should develop these new therapies in the clinic, and how we should collect data in order to answer some of these questions in the near future.